still allowed to say good morning, I hope. So, good morning. It's a pleasure to see you here. And I guess that this introduction was meant to give you some perspective of neuroanatomy. But from what I heard from you, most of you know a lot of neuroanatomy already. So, my name is Trygve Lergaard. I am a professor of anatomy, working in neuroanatomy, and also contributing to the Atlas development in the Human Brain Project. And the background has already been presented. So what I wanted to do was to kind of wind back and take the neuroanatomy perspective from a student's view. So medical students, they learn neuroanatomy by looking at the brain, learning that there is gray matter, white matter. There are landmarks, ventricles, pathways, and so on. There are textbooks, and there are, of course, also anatomical atlases, which simply give you plates and views of different structures that we can use to navigate with. And then, for example, you can do gross anatomy, and you can point to certain regions. For example, here there is black pigmentation somewhere in the brain. And you can learn that this is the substantia nigra due to certain cells that are there. And this is how most people are introduced to neuroanatomy. Okay. As we more and more work with radiological methods, it's becoming more important to also work with sections. And that is also introduced more and more in medical curricula, where you can slice through and look how different parts of the brain appear different according to which orientation plane you're looking at. Microscopes then, of course, can be used to look at certain parts of the brain, and you can look at details with different types of staining, such as you see here, as a Nissl stain and a myelin stain that allow you to perceive fibers in white matter and cells with different sizes and different densities and organization patterns in the gray matter. So, but that's kind of neuroanatomy at large. Students in medicine, they learn about systems and how they work and how they, through lesions and disease, can stop working, and that will correlate with symptoms. And that's about it. So, given the complexity of the brain, the anatomical atlases are not sufficient. So we also have brain atlases, and all of you working with science projects need to relate to atlases. And in that context, it's interesting to just stop and think about what is an atlas. An atlas can be anything. So an atlas is a collection of maps, images, illustrations, tables, put together to show you, inform you about something. Geography, feature, birds, fish, you name it. Brain atlases have a long history, so descriptions of the brain, they go way back. This is an example from Egypt. Textual description, here we have some drawings. And of course, brain mapping and brain atlasing really moved forward with the emergence of the microscope, the microtome, and different chemicals and procedures to stain specific features in the tissue. This allowed, for example, Corbini and Brodmann to describe certain patterns of cells in the cerebral cortex, which then allowed him also to subdivide these into different distinct areas based on the pattern of cells that he could see there. The Brodmann map of the cerebral cortex is, of course, famous, and here we also have this converted into 3D. So this kind of gives an outlay of various areas in the brain that have certain boundaries. And you can look at the map and then say, well, this is the distribution these areas have. In order to navigate this, you need coordinate systems. So this was already available many hundred years ago. You had plates with rulers allowing you to say, well, this is so and so far from this and that position. A coordinate system requires that you have some sort of coordinate grid. It needs to be defined with respect to where it starts and where it stops, units and so on. And this has been a very important approach to navigate to certain parts of the brain, in particular in surgery. So the stereotaxic coordinate systems have been developed to kind of help out with the need to navigate to a small spot in a complexly organized brain with instruments. And then there are many systems there. And such coordinate systems have then been applied to a variety of atlases. For the human brain, there are several atlases available. This list is not exhaustive. The font is small, but you may still recognize some of them. And we will hear more about the big brain and some of the human brain atlases that the Human Brain Project is using a little bit later, so I will not say much about it. Looking to atlases for experimental animals. I mean, rodents are used a lot, mice, rats, but also more animals. If you look at the number of atlases you can look up, it's a virtual jungle out there. There are many mouse brain atlases, many rat brain atlases. And of course, a question is, which atlas should I use? Which one is authoritative, which one is not? And the problem is that the brain is complex. So you have more than 11,000 neuroanatomical terms, and they are used in a very different way. So one part of the brain, one structure area may have several different names. Some researchers or experts may take the same term, but have different meaning attached to it. And there is, of course, individual variation in how the brain is organized. This varies also with age. And that makes neuroanatomy quite complex. So it's firmly established that experts disagree about how to deal with this. And I think that is a situation that will persist for many years. So this is a problem we basically have to deal with. And another problem also is when you read research data is that documentation often is missing. So that people say, well, we stuck our electrode into the CA3 region of the hippocampus, but they fail to report which atlas they have consulted or how they made sure that this was indeed the target they hit. And of course, stereotaxic atlases are a way to navigate in a reproducible way, and it has a long history. So the combination of detailed atlas diagrams related to coordinate systems was, for the rat brain, the atlas published by Paxinos and Watson was very important. And this atlas has, of course, as already was mentioned, it's widely used. It has more than 30,000 citations in the 30 years it's been around, and that really gives it some authority. But there are also some problems with it, and that is that, the plates are fixed, limited to a certain resolution, to certain spaces, and some of the boundaries have dotted lines, and there is no information explaining why the boundary was put here and not a little bit to the side or somewhere else. So the missing criteria is a big problem. But again, the value of an atlas like this is that it allows researchers to quite reproducibly navigate to a landmark, make a hole, insert an electrode or something else, do an experimental manipulation, and it's quite consistent across animals. In particular in mice, as far as I've heard. So, yeah. But the problem with criteria is one thing that one can address. We did a project on this some years ago, where we consulted Menno Witter, who's an expert in the field. He received a lot of requests to help out. I have my electrode. Here is a picture of my section. This is where I put my electrode. Can you tell me if I was in this region or in that region? And he helped out as best as he could, but he didn't have the time to do this properly. So one thing that for him was important was to try to document the criteria used to distinguish between different areas. There is a lot of literature about this. For the cerebral cortex, there are several book chapters describing carefully how you can distinguish these regions. For the hippocampus, we created an atlas resource that combines high-resolution images with criteria that you can use to navigate. Looks like this, so you can browse. So if you have a section and you wonder where is this region here, or how do I find the boundary of CA1 and CA2, for example, or CA2, then you can say, well, this is my experimental section. Here you can look up and find the corresponding level, but you can also read about the criteria that distinguish different regions. For example, if you're looking for the CA2, CA1 boundary, it's a drop in the size of neurons that's important. And here you have another characteristic for a different part. So here, this was one approach to combine this type of information. Yeah, I'll skip this one. But again, this atlasing approach was also limited to sectional material. And the Voxon Space initiative, also from INCF, was to move towards open access atlases that are based on high-resolution anatomical templates in which a coordinate system can be applied based on internal landmarks independent of the skull, because the skull is mostly removed when you work with experimental brain material. So the Voxon Space atlases for the mouse and the rat brain appeared. The rat brain atlas was based on a high-resolution MRI template, and here the anatomical signature in these different types of images was interpreted and used as a basis for anatomical delineations. And this has been done for several systems. And at the moment, there are the last version, so we have then a general version of the atlas taking the main brain regions, 2 more iterations of it, adding hippocampal structures, and lately now this year, auditory structures that have been interpreted on the basis of the MRI and DTI images that you see here, together with explanations and criteria on how these boundaries were identified. So, the main point I wanted to make here was that the 3-dimensional atlases, such as the Voxon Space Atlas, such as the Allen Mouse Brain Atlas, have the advantage that they are open, anyone can download them and use them. They are 3-dimensional, which means that you can use different tools to create customized sections through them that are independent of the standard principal planes. And then you can say, for example, in this example, we have an electrode into a brain. You see here that the slice is asymmetric. And anyone who has used or worked with small brains, used razor blades to cut them, may discover that, well, it's not always easy to reproduce the section plane. So this can vary. And therefore, the ability to take a 3-dimensional atlas, cut it to see the same landmarks, makes it much more easy to extract this particular location. So then you can define positions in the 3-dimensional atlas. And this is exactly what the Human Brain Project now strives to do, to assign location parameters to a wide variety of experimental data, so that it will be easier to find, view, analyze, and work with these data. And the main point is that and as Camilla showed earlier, the key to assigning location is what is the documentation that you have. What is the documentation provided from the researcher? What is the documentation you have in the lab? And the important point is, and then you can use this to describe location either semantically by saying, well, I'm here in this atlas. According to the atlas, and you should give a reference to which atlas you use, I am in this particular region. And then you can say that, well, my data is valid for this little region of the brain. Even better, if you have images, then you can align your image to the atlas, and you can extract point coordinates that pinpoint your data exactly in this particular atlas space. And that will actually allow you to say that if you apply a given delineation, then you have also a name for your structure. What you will, so this is how we work with heterogeneous experimental data. I mean, data come in all shapes and forms. They are functional data, structural data. Some come with image documentation and some don't. What we want to show you is that it is quite easy with the Quickly tool that you will be exposed to a little bit later, to take an image and map it into a 3D position. And actually, if you take a cell phone image of a brain slice you have dissected, it is quite feasible to put it into the atlas, find exactly where it is, and then you can document your data very precisely.